Okay, I think we'll start now. Welcome to day 2 of this workshop. Today we'll be discussing the principles of RNA sequencing data analysis. First of all, am I audible to everyone? Please just say yes if I am audible. Yes, okay. Okay. Before starting, I would like just to have a quick recap on yesterday's lecture. Let me share the screen. Okay, is the screen visible to everyone? Okay.
Yes.
I'm just gonna have a quick recap over yesterday's lecture. We have defined gene expression as producing gene products, the production of RNA and proteins from DNA during the process of transcription and translation. We have said that cell specialization and differentiation is brought by changes in gene expression and is brought by differential gene expression in general. There are multiple ways to measure gene expression. We have focused on RNA sequencing, which is a non-specific way to measure gene expression. We have defined RNA sequencing as a method of measuring gene expression by quantifying the amount of RNA that is transcribed from actively expressed genes in the genome. And we have seen the difference between the different types of RNA sequencing, including the single-cell RNA sequencing and the bulk RNA sequencing, and what are the advantages of single-cell RNA sequencing over bulk RNA sequencing. We have looked at the outline of bulk RNA sequencing, starting from RNA extraction into sequencing using next-generation sequencing, quality control, mapping the reads or fragments into the reference genome, quantifying the fragments that have been mapped into particular genes within the reference genome, and differential gene expression. So this is the RNA extraction, sequencing, the steps of sequencing, which starts with creating a cDNA library, and so on. We have looked at the different file formats that are used to store sequencing, one of which is FASTQ file format that's used to store sequencing and report the quality per each nucleotide or per each base. And we have talked about how to calculate the FASTQ quality scores, in which we have a Q value that's mapped into a symbol or a character using the ASCII system. Quality control, we have said that we should monitor our quality per base and we should trim any sequences with a low quality. And after a couple of minutes, we will see some of the tools that are involved in quality control. We have talked about mapping and aligning the reads into the genome. Any fragments that we have sequenced, they must be mapped into genes within the reference genome. And we have said that we have two types of alignment or sequencing: paired-end sequencing via single-end sequencing. Paired-end sequencing. During paired-end sequencing, we have a fragment that is divided into two ends. One end is sequenced in the forward direction, and the other end is sequenced in the backward direction. When referring to the file structures, in the case of paired-end sequencing, if we have, for example, two samples, or we are comparing the gene expression of two samples, we will have two individual FASTQ files for each sample, one referring to the forward reads and the other referring to the backward reads. We have said that all of the alignment that are stored in the sequence alignment map, the SAM or the BAM files, following the SAM and BAM file production, we should quantify the fragments that have been mapped into the reference genome. Quantifying algorithms need an input like the BAM file as well as the GFF or the GTF files. This is an example structure of the GTF and the GFF file. Yes. We have also talked about the importance of raw data normalization. Normalization takes into account the length of each feature or exon. It also takes into account the number of mappable reads on each feature within the reference genome. So normalizing the data would ensure fair comparison between the gene expression values. Transforming cell normalized data also to reduce the skewness of the data is also an important step. We have talked about differential gene expression analysis, how to determine whether a gene is significantly or statistically significantly differentially expressed between two samples. Differential gene expression takes into account the log fold changes, the magnitude of the log fold change, as well as the intra-group dispersion. So we have gene A is differentially expressed between the two samples, while gene B here is not differentially expressed between the two samples due to the high intra-group dispersion. And we have discussed this in details. We have talked about the main workflow of the DSEQ2 library, estimating the size factors for normalization, estimating the gene-wise dispersion, fitting a curve. After estimating the dispersion, the DSEQ2 must take into account the log2 fold changes. This is followed by hypothesis testing to give a p-value for each particular gene. So we can test for the statistical significance of each particular gene and whether it is differentially expressed or not. We have talked about the diagnostic plots that are used, including the MA plot. Genes with a higher normalized mean counts or expression values are more likely to have a greater magnitude of log2 fold changes, as you can see here, and they are more likely to be more significant. We have also seen the dispersion plots in which there is a inverse relation between the mean of normalized counts or the expression value of particular gene and its intra-group dispersion. We have talked about the different hypothesis tests used, including the Wald test and the likelihood ratio test to calculate a p-value for each particular gene. And these tests are linked to the chi-squared statistical testing. Following differential gene expression, we have talked about gene ontology enrichment and how we can link our list of the top differentially expressed genes to produce or to know the biological pathways that are involved in our analysis by linking to the gene ontology database. And the various methods that are used, including the ORA, FCS, and PTB. Okay. We have talked about the website, the SRA NCBI, to download the RNA sequencing data, as well as the Ensemble genome browser that's used to download the GTF files, the FASTQ files, and the GFF files. Yeah. So now, starting with the analysis, first of all, we should create a working directory containing all the files. So as you can see here in my computer, I have a working directory, RNA sequencing. I have all of the FASTQ files, the 4 FASTQ files in the case of paired and sequencing. As you can see here, we have 2 samples. We have the sample ending with 96, and we have the sample ending with 98. We have 2 individual FASTQ files for each sample, R1 and R2. These samples are taken from yeast cells that have been subjected to different environmental conditions or different kinds of environmental stress. So yeah, this is sample 1 and this is sample 2. Read 1, read 2, read 1, read 2 for each sample. In our working directory, we have also the reference genome of the yeast that we are studying. This yeast is known as Saccharomyces cerevisiae. It is a species of yeast which is known as the baker's yeast. I have chosen this organism since it has a small genome, so our algorithms will not take a lot of time to process. We have also the GTF file of the Saccharomyces yeast. We have the GTF file. Also, I have included a counts file, a ready counts file, an RNA sequencing counts file that shows the difference or the differential gene expression between stage 1 and stage 2 renal cell carcinoma. So our data analysis will be divided into 2 segments. Firstly, we will be studying the yeast. Secondly, we will be studying the differential gene expression between stage 1 and stage 2 renal cell carcinoma. since this will be a better example and a more real-life example than using yeast. So yeah, so this is the working directory. You should put all of the files in one folder, and you are good to go, including the script that I have provided in the drive folder. Okay. I'm just making sure that I'm recording. Yes, I'm recording. Now, before starting with the analysis, I would like to show an important tool that's used for quality control. We have a web-based tool known as Galaxy, or the Galaxy Genome Tool. This tool is used for quality control, for trimming, and many other procedures in bioinformatics. It's very useful. In this case, I have a sample FastView file. As you can see, we can view here the FastView file. Okay. You can see here the FastView file. We have our sample FastView file. By the way, if you want to use this tool, you must log in and have an account, and then you can use it properly. So we have this FastView file. I have ran a quality monitoring or a quality control on this FastView file. It's very simple. You just have to click on the FastQ quality control. We will use the FastQC tool that we've talked about yesterday. We will choose the file. In this case, this is our FastView file. And you can click on execute. It will take a couple of minutes, and it will display the results. As you can see here, we have a web page showing all of the quality parameters. So we can view the web page, and here is it. We can see here basic statistics about the sequence or about the FastView files. We have a total of 4,437 sequences. We have the pair base sequence quality, for example. The file is quite bad in terms of the quality, as you can see. Yeah, it's very bad. We have the pair sequence quality scores, the quality score for each sequence. The GC content also, the guanine, cytosine contents across the sequence, and so on. So we have multiple parameters, and you can view them in depth. So yeah, this is just the main idea of FastQC tool. It's very simple to use. Also, if we want to trim the low quality sequences, we can use a tool such as Trimomatic, or even we have here the Trim Galore tool. This is a popular tool that's used for trimming low quality sequences. So you can click on this tool. You can choose the file. For example, this is our file. This is our file that we want to control its quality. By clicking on advanced settings and clicking on the full parameter list, we can choose the threshold for the quality value. In this case, the general quality of our file, as you can see here, the general quality is low. So we can't specify a threshold value of 20, since most of the file will be removed. So we should have a balance. I think, for example, here choosing a quality score or a threshold quality score of 8 would be a good option. So by returning to the Trim Galore tool, advanced settings, full parameters list, we can choose, for example, a threshold value of 8. And by executing, we will have the new trimmed FastQ file, as you can see here. And we can view it. This is the new trimmed FastQ file. Okay. Oh, here is it. So yeah, this is the new trimmed FastView file. After running the Trim Galore algorithm, we are able to trim any sequences or any bases with a quality score that is less than 8. By the way, you can learn more about all of the tools that are present in the Galaxy Genome tool. It's a very useful tool in bioinformatics. So make sure to use it today and try running quality control on multiple FASTQ files. If you want to run on FASTQ files or sample FASTQ files, you can type on the internet FASTQ sample files, for example. Yeah, you can use this website, for example. You can download a file with a low storage capacity, for example, this file, and you can run or try quality control on it. So yeah, I think this is enough for the Galaxy Genome tool. Now let's start with our analysis. We'll be using mainly RStudio. RStudio for R programming. It is the most popular IDE for R. Okay. We'll first make sure to set our working directory. As you can see here, this is my working directory. Please make sure to have double backslashes when specifying your path. This is better when using the RStudio. So we can set the working directory. As you can see here, we have our FASTQ files. Yeah, we have all of the files, the reference genome, the counts file, and so on. You should start by installing the required packages or libraries. We have the normal libraries, such as the ggplot library that we'll use for plotting. So you can install it in this way. The r.utils library also. And by the way, I have provided the R script in the drive folder. We have some libraries or packages that are related to the bioconductor manager or the bioconductor framework. To install these libraries, you should execute these 3 lines of code by removing the hash, surely. Yeah, you should execute these 3 lines of code to download it properly. Yeah, and just click enter. I have the libraries or the packages already installed, so I don't need to run these 3 lines. Okay. We have also the data-to-table library that we'll use for the mapping, quantification, and the variant analysis. In this lecture or analysis, we'll not refer to any kind of variant analysis. Yeah, we'll just map and quantify the reads. Variant analysis is another topic for whole genome sequencing. Okay, here is another bioconductor-related package. Also, it's the same, the RNA-Seq package that we'll use to remove unwanted variation from RNA sequencing data. Also, we have another bioconductor package, which is the DSEQ2 for the differential gene expression. We have also the enhanced volcano package that we'll use to produce a special kind of plots known as volcano plots, which are very good at visualizing differential gene expression. We'll use the normal library, the R-Color Brewer library, to specify our color scheme for the plots. We'll use the p-heatmap also library to produce the heat map plots. P-heatmap refers to pretty heat maps, so yeah, and they are actually pretty heat maps. Okay. We have another bioconductor manager library, which is known as the RQC. By the way, you don't need to install the RQC. The RQC is just for quality control. So you are free to choose between RQC or Galaxy or the FastQC tool. RQC is also a good tool, but it takes a lot of time. So yeah, you can install the RQC as you like. Now, to start, we should make sure that we have our reference genome, the FASTQ files, and the GTF files in the same directory. So I think, yeah, we are good to go. We can start by running the quality check by executing the RQC function. The parameters for the RQC function, we will specify the path. In this case, this is my working directory or path. And we will specify the files that we want to run quality control on. In this case, we will choose all the files that have the following pattern: .fastq, .gz, zipped fastq files. So yeah, I'll not run the function now since it takes a lot of time. It takes around 15 minutes. However, after running this function, you will have a web page displayed for you like this, an HTML page containing all of the quality control related parameters for all of our files, our 4 files, as you can see here. We have a lot of parameters in the pair-based quality, basic statistics, the base calls, and so on. Okay. So after running the quality control and after running the quality check, we should start with our analysis now. Remember that one of the first steps to start the RNA sequencing process is to align our reads or fragments within our FASTView files into the reference genome. Before the alignment process, we must build an index for our reference genome. We must index our reference genome. What I mean by indexing, suppose, for example, you are searching for a word within a book. It is much better and much more efficient to use an indexed book to search for the word according to its alphabetical order, for example. It is the same for indexing the genome. We should index our genome. We should create indexes for each sequence within our reference genome in order to align the reads efficiently and align our fragments efficiently. So we can build an index to the reference genome by using this function, build index. We can specify this parameter, reference index, which is the name of our output file or the name of our index output. So you can name it reference index or whatever you want. We will specify our FASTA file or the reference genome file of the yeast, for example here. You can also set this parameter value to false, index split to false. What does it mean? Index split refers to splitting the index or the output index into blocks. In the case of this small genome, we don't need to split our index. However, in the case of larger genomes, like the human genome, you should split the index into blocks. And by the way, if you want to search for any function and to know more about any function, you can here specify in the console like this. For example, build index. As you can see here, build index, and we can know more about it. For example, here it says that an index needs to be built before read mapping can be performed. This function creates a hash table index. So it uses a hash table data structure to index the reference genome. If you don't know what are hash tables look like this. So each particular sequence will have an index within the data structure. It is a common and very popular structure in computer science. Okay, so after building the index, I will execute the function now in real time. It will take some time, I think, so we can wait. Okay, I hope it will not take a lot of time. I think 2 minutes. And by the way, it depends on the computational power of your personal computer. So yeah. Okay. Okay, I think it's running slow right now. if you have RStudio installed, you can, I think, start your data analysis right now. Okay. I'm not sure why this time it's taking an unexpected long time, but I can send you the links for the genome tools. Here is the link for the Galaxy genome tool in the chat box. I'm really sorry for waiting, but okay. By the way, before running this algorithm, you should check your memory usage because it takes a lot of memory. So I think I'm having a virtual machine turned on. So We should find a solution. By the way, when you install the packages, you will see it will take a bit of time. Okay, now it's running fast. Okay. As you can see, it's saving the current index block. And we are using only one block, so we're not splitting into blocks. 70%. Okay, now the build index is done. And as you can see in our working directory, we have the reference index stored and other accessory files that are used. Mainly, we have the reference index, and we'll be dealing with the reference index file. Okay, now we should collect our FASTQ files into variables, reads1 and reads2. Reads1, we will store in reads1 all of the FASTQ files with forward reads. In the case of paired end sequencing, all of the FASTQ files with this pattern, R1 for the forward read, will be collected into reads1. And all of the FASTQ files from both samples with R2 reads or backward reads will be collected into the variable reads2. So you can execute it. Reads1, reads2. As you can see here, reads1, by viewing reads1, we have R1 from both samples, the 96 sample and the 98 sample. Also, we have reads2, we have R2 from 96 and 98. Now we should start with the alignment process. The alignment process, we should use the align function from the R subread package. We should specify the index that we have produced, the reference index. The read files, we have, in the case of paired end sequencing, we will have two read files, read file 1 specifying the forward reads and read file 2 specifying the backward reads. The align function will be able to differentiate between both samples. It will be able to differentiate between both samples. We will specify the input format. In this case, we are using the FASTQ file format, the GZ FASTQ file format, or the zipped FASTQ file format. We will specify also the output format, which is the binary alignment map or the BAM file. Also, we can specify the number of threads or CPU threads. In this case, I'm using 10. It is by default 1. So yeah, I think 10 is suitable. It depends. Okay, we can execute the align It takes a bit of time also. This is why I didn't choose genomes like the human genome. The human genome would take a lot of processing time. So, yeah. Okay. If you have any questions, you can put them in the chat box and I'll answer them later. As you can see, the alignment algorithm is related to the RSubread package that we have installed earlier here, the RSubread library, which is a part of the bioconductor framework. Okay. Okay, so we have aligned the file successfully for now. As you can see here, we have the full report of the alignment and we are good to go. And if we check here, we have we check our working directory. We have our BAM files stored. So as you can see here, we have the BAM summary and we have our BAM files stored. As you can see here, we have the .bam extension, the .bam extension, and so on. We can list our BAM files using this function. So as you see, we have two BAM files for both samples, 96 and 98. Okay. I'll return in 1 minute just. Okay, so as you can see here, we have our BAM files, and now we are good to go. We can check the quality, the mapping quality of our BAM files by using the prop map function, and we can specify the files, the bam.files variable. So, yeah. Okay, we can view the props. As you can see here, we have the total number of sequences and we have the number mapped. It's quite difficult to sequence all or to map or to align all of the sequences. So yeah, the alignment has a certain quality. As you can see here, the probability mapped, here we have around 97%, here we have around 98%. So yeah, it varies. But overall, it's good. Okay. Now, after aligning our fragments or reads to the reference genome, we must start with quantifying the number of fragments or reads that have been mapped to each particular gene within the reference genome. We will use the feature counts library or the feature counts function. For the feature counts function, we will store the object or the counts within a variable known as fclim. You can name the variable whatever you want, but for now it is fclim. We can execute the feature counts function. The inputs for this function, we will have the BAM files that we have saved into the bam.files variable earlier. And we will use the annotation GTF file that we have downloaded previously that is specific to the Saccharomyces cerevisiae genome. We are focusing on exons primarily. We are focusing on coding regions within our DNA. We are not focusing on non-coding RNA. We are just focusing on exons. So we'll specify the feature type because the GTF file contains information about exons. It contains information about non-coding regions. Yeah, it contains information about the whole genome. So we must specify the feature type, in this case, exons, since we are dealing with gene expression. Okay, we must also specify the attribute type. We are mostly dealing with gene IDs. Each gene has an ID, so we are dealing with the gene, common gene IDs. Yeah, and we are here making sure that we are using a GTF annotation file, and we are making sure that we are using paired end sequencing. If we are using single end sequencing, we would specify this as false. However, in this case, we are using paired end sequencing, so its value will be true. Now we can execute the feature counts function. Okay. We can convert our fclim. We can extract all of the counts from our fclim object using this indexing procedure, as you can see here. If you are familiar with the R programming, or if you are familiar with the R syntax, then we are extracting the counts from the fclim object, and we are converting these counts into a data frame. Okay, and storing the data frame in a variable known as fc. We can specify the column names of fc, for example, sample1 and sample2, or for anything you want. In this case, sample1 and sample2. We can view fc, and as you can see here, we have produced our counts file and gene expression data from our original FASTView file. So you can see here the gene IDs. We have 7,000 genes, and we have both samples. Okay, so this is the main idea of producing a counts file from FASTView files. Okay. Here there is a question why you choose only exon feature. Please, what is the objective behind it? Basically, in our analysis, we are just focusing on regions within the DNA that are coding for protein production. So we are focusing just on exons. We are not analyzing non-coding regions of the DNA. We are not analyzing introns. We are just focusing on exons. So yeah, this is just the main goal behind choosing the exon feature. Okay. Now we have worked on sequence alignment and fragment quantification. Now let's work on a more real-life example, or on a better example. We'll be working with a human renal cell carcinoma samples. We'll be working on or comparing the gene expression and carrying out differential gene expression between stage 1 and stage 2 renal cell carcinoma taken from humans. However, we'll not do these steps. We'll not do the alignment process, and we'll not do the quantification process. We have a ready quantification file here. We have a ready counts file. This is our counts file. So you can suppose that we have ran the alignment process. You can suppose or assume that we have quantified our fragments, and now we have this counts file. The main reason why I'm not working with alignment with the human genome, as I said earlier, that it will take a lot of time and it will take a lot of storage, and not everybody will be able to run the analysis on any computer. So yeah, we can start with a ready counts file. It is a text file. We can read it as a CSV, as you can see here, with a separator, a space separator, since the values are separated by spaces. And we can specify that there exist row names. Okay. So we can read our text file. We can view. Okay. Now this is the main structure of our counts file or text file. Yes, exactly. Exon region code for protein only. Yes. Okay. So we will have the row names, which are the IDs of the genes that we are studying. I have shown the head only, just the first 6 genes. However, we have a lot of genes. If we view we have 57,358 genes. However, here I'm just showing the first 6 genes. As you can see here, we have the rows specifying the gene IDs, and we have the columns specifying the replicate samples. T1 refers to stage 1 renal cell carcinoma, and we have T2 referring to stage 2 renal cell carcinoma, and we have replicate samples of each stage of renal cell carcinoma. As you can see here, we have 24 replicates of T1, and we have 26 replicates of T2. So we can start our differential gene expression analysis. We first should count the number of T1 replicates and the number of T2 replicates. Okay, sorry, here it's T2 count. It's T2 count, it's not T1. So starting with stage 1, as well as, okay, no, we can view, we have 24 replicates of stage 1 renal cell carcinoma. Okay, we can count stage 2, and we have 20 replicates. Okay, it's 20. Okay. If you count them, they will be 20, yes. Now, after counting the number of replicates for each sample, stage 1 and stage 2 renal cell carcinoma, we can start with the filtering process. We would like to extract the genes with an expression value greater than 0 in at least 2 samples, or in at least for 2 replicates. We can say 2 replicates out of the 44 replicates. So yeah, we want genes that have an expression value greater than 0 in at least 2 replicates, or in at least 2 columns out of the 44 columns. So we can run this function. This function, if you are similar with, for example, the Python syntax, this corresponds to the lamb function in Python. While here you can put a function as a parameter for another function. In this case, we are using the apply function, and we are putting an anonymous function as an argument. So we will run the filtering process or the filtration process on our data structure or data frame if we will specify that we want to filter the rows. We are filtering genes, and genes exist as rows. So if we return to our data structure, as you can see here, we will be dealing with the rows. We are filtering the rows. So we will put 1 here. Okay, and we will define our function as an argument. The function will be giving a binary output, a 1 or a 0. If the following, it will be giving the length of the array that contains a binary output for the cells that contain a value greater than the expression 2 or greater than the value 2. So what I mean by this, we can run the function here. We can save the data from these text files. Yes, we can save to Microsoft Excel, and we will do this later. Yes. So yeah, we'll run the filtration process. The filtration process will generate, it will generate for us indexes, as you can see here. So we can index our data frame again using the output filter. and store it in a variable filter, in this case. So filter, as you can see here, it contains around 37,992 genes, which are the filtered genes with an expression value greater than zero in at least 2 replicates out of the 44 replicates. So we are done with the filtering process. We can store all of our gene names within the variable genes also. All of the filtered gene names in our variable genes, and we are good to go. Okay. So now we should define variables T1 and T2 since we are dealing with stage 1 and stage 2. Okay, T1, it will be a vector of the following string T1 replicated ST count times. So it will be replicated, for example, 24 times. If ST1 count is equal to 24, let me check. It's equal to 24. So we will have a vector containing the T1 string replicated 24 times by using the replicating function, and the same for T2. So if we view T1, as you can see here, this is T1. As you can see here, this is T2. We will use this for the annotation later. And we will join both vectors, T1 and T2, into one vector called X. Okay, if you view X, we have joined them together and concatenate them together. We can use also the factor function to divide them into levels. So this factor function will be able to divide T1 and T2 into 2 levels. And if you view X here, as you can see, we have 2 levels, T1 and T2. We should divide them since they are both separate samples, and we are running differential gene expression between the 2 samples. Okay. Yeah, after we have prepared our annotation variables, we should start with creating an expression set object. The expression set object is usually a digital file format that's used to store RNA sequencing data, and it's used to store RNA sequencing counts data. It's an efficient object. So yeah, we'll define a set as the new set expression object, or the new sequencing expression set object. The counts of our object will be the matrix filtered. Remember here, we have filtered our counts file and extracted the genes with expression value greater than 0 in at least 2 replicates. So this is the counts of our object. We will also specify the pheno data, which are the representation data or the annotation data. We will specify the variable X that we have created for the annotation of the stage of the renal cell carcinoma, and the row names. We will specify the row names. The row names will become the column names of filter. So as you can see here, we have the column names of filter, T1 and so on, specifying the stage of the renal cell carcinoma and the number of the replicate. So we will make the row names of this set to be the column names of the filtered object, or of the filtered matrix. And we will see later its structure. Okay, we can create set. I couldn't find function. Okay, I think it's just. I think one of the packages are missing. Yeah, I have one of the packages missing, I think. So yeah. Okay, we're now good to go. We can include this package right here. And I will send you the modified script file and share it in the drive. Okay. By the way, when working with data analysis, you are subjected to many errors. So I highly recommend you searching on the internet for the error, and you can find solutions. Programming is quite stressful. So yeah. So we have created our new expression set object. We can view our set. As you can see, our set, it contains the features, the 37,992 filter genes, 44 samples, including the samples and the replicates. We have the counts, we have the pheno data, including the variable X, as we said before, and so on. So we can start with the analysis. We can set the color scheme of our plots using the color brewer function. Yeah, I have chosen here the set 3. You can choose the set that you like. For example, set 2, it's just about colors. So we can choose our color scheme. Remember that we have our new expression set object containing our counts file and containing our data annotation fully. We can start with a plotting basic graphs before starting with our normalization procedure. One of the plots is known as the relative log expression plot. It is just a collection of box plots for each particular replicate, as you can see here. For each replicate, we have a box plot specifying the RNA-Seq counts values. However, it uses the logarithm of each value. So it runs a logarithmic calculation, and then it displays the box plot of the values after the logarithmic transformation. We can also, for example, cluster our samples or replicates using algorithms like the PCA algorithm or the principal component access algorithm. In our data, we have a lot of genes. We have 37,000 genes. So we have a lot of dimensions, and we cannot see 37,000 dimensions. We can see up to 3 dimensions only. So we usually resort to techniques like PCA to reduce our dimension from, for example, 37K dimensions into 2 dimensions. So we can cluster our replicates or samples according to their gene expression profile in a 2-dimensional space. So if we plot it here, we will specify the set that we are using, our object that we have created. We have also specified the color scheme and the size of the points, and so on. As you can see here, we have clustered our samples or replicates, from both samples, stage 1 and stage 2, according to their gene expression profile by using only 2 dimensions and not 37,000 dimensions. So yeah, however, it's unlikely to preserve all of the variation within our data when reducing our dimensions. So as you can see here, our first principal component just contains 27.5% of the variation, and PC2 contains only 13.98% of our variation. So yeah, it's impossible to preserve 100% of our variation after dimension reduction, but still it is a useful technique. Other dimension reduction techniques and clustering techniques exist, like the TSNE and the UMAP algorithm. These are mostly used with the single-cell RNA sequencing and not the bulk RNA sequencing. Yeah. Okay. Now, we should normalize the data. In this case, we are using the upper quartile normalization. So we will redefine our set we will run this normalization function. In this case, it's called between lane normalization. This uses upper quartiles to normalize the data. You can look for upper quartile normalization on the internet, so you can learn more about it from a statistical side. Yeah. So we are running this function on our previous set, the unnormalized set, and we are resaving the normalized set into a variable called set again. So yeah, and we are specifying that we are using the upper quartile and not the lower quartile here, or not the median, or so on. So we run the normalization. Okay, we can plot now after normalization the same plots, the relative log expression plot, as you can see. If you compare it with the previous one, you will see that the variation has been reduced. For a better example, you can see here an RLE plot before normalization, and we have here a plot after normalization. As you can see here, before normalization, after normalization, the variation has been reduced and unwanted variation has been removed. Yeah. Okay. We can also run the plot PCA or the clustering. As you can see here, the values are closer to each other and we have reduced the variation as opposed to our previous unnormalized plot. The values are quite more spread. So yeah. Okay. Now we should divide the stage 1 and stage 2, T1 and T2 samples replicates into 2 groups. This function is a part of the RNA-Seq library. This is used for the differential gene expression. So we are now really dividing the samples into groups to start the differential gene expression. And we can store these groups into a variable called differences. Okay. I think I have a problem with the RNA-Seq library. It's installing the package right now. Let me try. Okay. Okay, I'm gonna update them. Okay, let me try. Yeah, I think we are now good to go. I have included the RNA-Seq library. We try to make the groups. Yeah, it's now working. If we want to check or view the differences variable, as you can see, it has divided into, if we can view it, it's better. It has divided the samples into arbitrary groups 1 and 2, stage 1, stage 2, and stage 1. As you can see here, we have -1 values specifying that there are no columns for stage 2. The number of stage 2 replicates are less than the number of stage 1 replicates by 4 columns. So these columns will show as -1 in this case, since they are not present and they have no value. Okay. Now we can start with reducing the unwanted variation by using the rovs function. So we can define a set3, for example. You can name it set3. We can run the rovs function, which removes any unwanted variation. We can run it on the set, on our set, the expression set. We can specify our genes that we have extracted. Remember before, we have extracted our filter genes. We must also specify the number of unwanted variation factors to be calculated. In this case, we are calculating 1 for each gene. We are calculating a factor, a size factor that's used for removing the unwanted variation. By the way, I have provided the documentation for the RNA-Seq library in the Drive folder. If you want to know the details, you should read the documentation. Reading documentations is extremely important. Okay. And we are specifying the groups, which is the differences variable that we have created. Okay. If we view these are our the p data of the set 3, or the factors that we are using to reduce or remove the unwanted variation. Okay. The documentation here, it contains, as you can see, a whole section for estimating the factors of unwanted variation using replicate samples. So yeah, you can have a much better understanding by reading it. But it quite needs a background in linear algebra. The RNA-Seq library highly depends on linear algebraic principles. So yeah, you should ensure that you have a good mathematical background before understanding its main way of working. Okay. Now, after reducing and removing or eliminating the unwanted variation, after normalizing our data, after defining our counts set expression object, now it's time for using the DSEQ library, and it's time for the differential gene expression. So we can create an object, a DSEQ data set from matrix. This is an object, a differential gene expression object that we create. This object is created by using the DSEQ2 library, and we will store it in the variable known as DDS. We will specify the count data, counts of set 3, which is our expression set object with unwanted variation removed and with normalized values. We will specify our column data, in this case, our P data, the variation factors, and our P values. We will also specify the design. The design refers to the type of hypothesis testing that we'll be using. In this case, we will be using the Wald test and the likelihood ratio test. This is the syntax that is used usually to specify the Wald test and the likelihood ratio test. And we should also specify the X, which contains our replicate samples, our factored replicate samples here. With divided levels. We have the T1 and T2. The main idea of differential gene expression is to compare the expression values between two samples. So we should provide an annotation variable specifying the levels, T1 and T2 at least. Okay. So we can define DDS now. You will see now how we can run the DSEQ analysis now on the DDS set matrix by using the DSEQ function. You can see that now it is estimating the size factors. As we said in yesterday's lecture, it is estimating the gene-wise dispersions. If you remember here, It usually starts with estimating the size factors, the gene-wise dispersion, fitting the curve, and so on. So now it is estimating the gene-wise dispersion. It takes a bit of time. It is finding the mean dispersion relationship, which is the MA plot here. The normalized mean, the dispersion plot, the mean of normalized counts and the dispersion. The final dispersion estimates, fitting the model and fitting the curve, fitting the dispersion estimates into the expected dispersion values and shrinking them. If you don't understand the details of RNA-Seq2, I have also provided the documentation for it, and you can read from it. Okay, now we are done with the RNA-Seq analysis. We should store the results of our RNA-Seq analysis in a results variable and using the results function. So, yeah, we can review the results. As you can see, if we view it, okay, it's an object, so we should view it like that. The results, we are having a set of genes. We are having the log2 fold change for each particular gene, the mean for each particular gene. We are having the p-value and the p-adjusted value. As we said yesterday, we will be having a list of genes with the adjusted p-value, the p-value, the log2 fold change, and so on, as well as the gene ID. So these are the results. We can have an MA plot on the results, which is a diagnostic plot. Okay, I think. Okay, for example, here we have our dispersion plot. As you can see, it is indicating good values. We are having an inverse relation between the mean of normalized counts versus the dispersion. So yeah, it's a good diagnostic procedure. I think there is. Okay. Okay, for now we are good with the dispersion. We can write all of our results into a CSV file. For example, here the RNA-Seq analysis CSV file, and we should specify the separator here. For example, a comma separator, since we are dealing with CSV files. So yeah, we can write the results. If we check our working directory, we will have our CSV file containing the results of our differential gene expression analysis. As you can see here, the gene ID, the base mean, the log2 fold change, the p-values, the p-adjusted values, and so on. Okay. Now we can read our CSV file that we have stored in a variable known as data frame. We can filter this file to just extract genes with a p-adjusted value that is less than 0.05. So now we will be extracting the differentially expressed genes with a statistical significance. Okay. We can now start plotting after. As you can see here, the DF, this data frame will contain all of the statistically significant genes with a significant p-value. As you can see here, our p-values are significant and less than 0.05. Okay. We can plot a heat map for the first 4 genes. Here we have a heat map for the first 4 genes, A1BG, 7SK, and so on, across the replicates, as you can see. A more intense color means a higher expression value. And you can use the scale here. All of this work is done by the pHeatMap function, by the way. And here we are ensuring that we are scaling across the row. So we are visualizing relatively the gene expression relatively across all of the replicates on a relative scale. We can also produce an enhanced volcano plot using our results for the differential gene expression. Okay. We will run this enhanced volcano plot on all of the genes. The results variable that we have defined earlier, which contains the results of the DSEQ2 analysis. After having the plot, the enhanced volcano plot is very useful to show differential gene expression. Genes that are marked in red are usually statistically significant and they are differentially expressed. As you can see here, this gene is highly differentially expressed. This gene is highly differentially expressed. The genes in the middle are usually not significant. And usually the significance is determined by what? It is determined by the p-value and the log2 fold change together. And the dispersion, as well as the dispersion during our data analysis. Okay, as you can see here, for example, this gene is also highly differentially expressed. So yeah. If we look here. Sometimes our enhanced volcano can look like this, which is the ideal type containing the differentially expressed genes here, and in the middle, the non-significant genes. These genes are usually down-regulated, and these genes are up-regulated. Okay. Now, for further plotting, we can transpose our matrix. So remember, just a minute. Remember, we have our original matrix, F, here, our CSV file containing the counts file. We can convert it into a data. We can firstly transpose it. So the transposition of the matrix, we will switch the row names with the column names and vice versa. So this is the transposition of a matrix. So if we have, for example, the row name, if we have the column names and we have the row names, for example, the row names representing the gene IDs and the column names representing the replicate samples, by transposing the matrix, we will invert the distribution of the row names and the column names with respect to the values, for sure. So yeah, by using the T function to transpose it. Okay. we can convert it into a data frame to work with, and we can view it, as you can see. Okay, I have transposed it 2 times. You can retranspose it. Okay, as you can see here, we have the replicate samples as the rows, and we have the gene IDs as the columns after the transposition. Now we can create a vector cancer stage. I have named it cancer stage. We can replicate the string T1 for ST1 count and string T2 for ST2 count. We have done this process earlier, but we will redo it now. Okay, if we as you can see here, we have a cancer stage. It is similar to the X variable that we have defined previously. Okay. Now we can bind this vector to our data matrix or counts file by using the cbind function or the column bind function, and we can view. As you can see here now, the cancer stage is a main column within our data structure or data frame. We can plot Violin plots and box plots. Violin plots, if we plot the Violin plot using the ggplot library, we will specify our data frame. We will specify our data annotation, including the replicate samples or the cancer stage annotation. We will specify the gene that we want to plot a Violin plot for it. In this case, I'm choosing, for example, the A1CF gene. We will specify the type of plot, the Violin plot, and the jitter. We are specifying the shape of the points and the shape of the points in general. So if we produce this Violin plot, okay. Here we are comparing or visualizing the differential gene expression or the difference in, or we are visualizing the counts expression values of the gene A1CF between T1 and T2 or stage 1 and stage 2 renal cell carcinoma by using a Violin plot. As you can see here, the difference in width or the width of the plot represents the density of the points here, or the density of the points plotted. Here we have a lower density. Here we have the greatest density, so we have the greatest width. We can deduce that A1CF has generally higher expression in stage 1 renal cell carcinoma than stage 2 renal cell carcinoma, since the white base is up here and the white base is down here. So yeah, it is very similar to the box plot. However, here it is showing the density of the points. Overall, it is showing a distribution. So yeah, but in a different way visually. We can also have a box plot for the desired genes. We can specify also the data matrix again. We can specify our annotation, a cancer stage, also for the same gene, A1CF. By the way, you can put any gene here, the name of any gene. Also, we can specify the type of the plot again, the box plot, and the same shape of the points. You are free with choosing the shape. So we can have our box plot. And as you can see here, this is a normal box plot showing the anomalies. We have the upper quartile, the lower quartile. We have the median, sorry. As you can see here, the median count of A1CF gene in stage 1 renal cell carcinoma is greater than in stage 2 renal cell carcinoma. So we can deduce that A1CF is downregulated during the transition from stage 1 into stage 2 renal cell carcinoma. So yeah, this is the general distribution. You can try with a lot of different genes. Okay, so this was the introduction to RNA sequencing data analysis. These are usually the main plots that are used. I have provided in the materials a recommended paper to read. As you can see here, this paper RNA-Seq of circulating tumor cells in stage 2, stage 3 breast cancer. As you can see here, the paper contains the volcano plots. It contains the clustering techniques, the principal component analyses. It contains the heat maps. Yeah, so you can read it. It has also pathway enrichment, enriching using gene ontology. So you can read this paper and know more. Reading papers is the best way to learn bioinformatics. And you can also train yourself by using the data sets that are provided as sources or supplement materials within each published paper or publication. Okay, here somebody's saying that I want to see the gene ontology. Would you please provide the piece of code that can do that? Gene ontology, as I said before, you can use tools, web-based tools. You can also use some functions in R. I have just a minute. You can use the web-based tool, or you can use in R a lot of gene ontology functions. As you can see here, you can use the enrich KEGG function to map for the KEGG pathways, and you can have a bar plot. You can use the GO enrichment, the GO biological pathways. You can see the link in the presentation and click on the link and use these functions, or you can use a web-based tool, as I said earlier, such as the ShinyGo tool, for example. Where you can provide a list of the differential expressed genes. Let me try now. Should have. We can take a list of genes from our Excel file, for example. Yeah, after submitting, it will show you the results, the gene ontology results. Saccharomyces. Okay, I think it has to download or retrieve the gene IDs for the Saccharomyces cerevisiae. Okay, and now it will show you charts. For example, here it has identified this pathway, for example. It can also link to the KEGG pathways. If you have a real list of top differentially expressed genes, it will show you all of the results. But for now, I think it's taking time to retrieve the gene IDs. However, it's very simple. You just have to put the list of genes and submit, and it will show you all of the results. I'll try to send you a list of genes on WhatsApp, for example, and you can try by yourself using this tool. Okay. You can also check the GitHub link that I have provided. So you can check also the R functions that are involved in gene ontology enrichment. But I'm not sure why it's not working now with me. I think we can try on demo genes here. Yeah, here, for example, we have tried with demo genes. It has shown all of the significant pathways. As you can see here, DNA damage checkpoint signaling, mitotic DNA damage checkpoint signaling, cell cycle checkpoint signaling, and so on. You can see the charts. Yeah. The KEGG network, you can link to it using other tools. You can see the interactome or the topology of the pathways. You can see that the more significant genes or the more significant pathways are showing as bright green, the less significant pathways are showing as pale green. You have a full tree for the pathways, phylogenetic tree, I think it's called phylogenetic. So yeah, this is the main idea behind gene enrichment. You can see here the number of genes, for example, that are involved in each pathway, and so on. Okay, I think we're done with the data analysis. I think this data analysis is good for a start in RNA sequencing and a start in bioinformatics. You will be able, I think, to read publications with a good flexibility, because these are the main plots that are usually used in differential gene expression analysis. Sometimes scientists try to customize their own plots, but these are the main templates or plot templates that are used: the Bob-Violin plots, enhanced volcano plots, heat maps, and so on. So yeah, I hope you like this lecture. Tomorrow we have a quiz, a mini quiz of 20 questions, or no, I think it's less than 20 questions. You should attend the quiz and receive a passing grade in order to get an e-certificate. The e-certificate will be sent to you by email a few days later. Regarding the recording of yesterday's lecture, it will be sent after a few days, since it needs time. So yeah, thank you. And yeah, any question? If there are any questions, you can. The timing of the quiz will be 1:30 p.m. GMT. I will share it. Okay, you are welcome. Okay, thank you. I will share it on WhatsApp, the timing of the recording.